This is the third and final video in Module 2, DNA replication. In this video, I'll be talking specifically about the enzymes and the process for DNA replication in cells that contain a nucleus, also called eukaryotes. Because eukaryotic genomes have a different structure and a different shape, eukaryotes have to replicate their DNA uniquely compared to prokaryotes. There are some overlaps, but there are still quite a few differences as well. Eukaryotic genomes are linear, and so they're a different shape. They're going to have problems replicating the ends, and I'm going to talk about that in a minute. Eukaryotic genomes also tend to be a lot larger, and so they have to take advantage of using multiple origin of replications in order to quickly replicate their genome. And so linear eukaryotic genomes contain thousands of origins where there's DNA synthesis takes place at multiple times within a single chromosome. Eventually, the replication forks will bump into each other and the DNA segments will fuse. And then we do have one issue where we're going to have trouble replicating the very ends of the linear chromosomes. So we'll come to that point later in this talk. Some of the requirements of replication are the same. So we need a template strand, we need nucleotides, we need enzymes and other proteins to facilitate the process. And this happens in a semi-conservative manner, just like it does in prokaryotes. So there's differences and there's similarities. So let's talk about the differences now. Eukaryotic DNA replication begins at the autonomously replicating sequences, or ARS. This is essentially an origin in a bacteria. Just call it something different. It's also a lot shorter. It's about 100 to 120 base pairs, whereas if you recall, the origin of replication in bacteria is 245 base pairs long. And these are the order of those nucleotides are species specific. And so an ARS in a human might look different than an ARS in a mouse. The origin recognition complex are the set of proteins that bind to the ARS to initiate DNA replication. And so the origin recognition complex is sort of like the initiator proteins in bacteria. The licensing of DNA replication, or the beginning, where it's the go-ahead, is initiated by the replication licensing factor, or the MCM. And this is basically the same as helicase that does the unwinding. And then there are specific eukaryotic DNA polymerases that do the elongating. So let's look at this step by step. Here's our DNA molecule to be replicated. And within there's some type of origin. The origin here, remember, is called an ARS, or an autonomously replicating sequence. The ORC, or the origin recognition complex, which is like an initiator protein, or the DNA E proteins in prokaryotes. So these are initiator proteins in prokaryotes. It's the same thing as the ORC in eukaryotes. They're DNA binding proteins, so they're going to bind to this DNA at this very specific site. And then it's going to recruit some other proteins to it. And then those proteins work together with the ORC to recruit the MCM complex, which is very much like the helicase in bacteria. And so the MCM is what's going to travel along and open up, keep the replication bubble opening at the two replication forks. Elongation proceeds in a bit of a different way, depending on which DNA, it proceeds in a slightly different way, only in the fact that there's so many DNA polymerases that can elongate DNA in a eukaryote. Many of them are involved in DNA repair. And so translation DNA synthesis is related to DNA repair. And so we have DNA repair, lots of DNA repair here. All of this is our DNA repair, our polymerases. We also have certain polymerases that are involved in nuclear DNA replication. So that would be this one, maybe involved in replicating the DNA in the nucleus. And those are distinct from the polymerases that replicate DNA in the mitochondria. Remember, mitochondria have their own DNA, their own genome. And so separate DNA polymerases are responsible for replicating that. But the two major DNA polymerases involved in elongation are the delta DNA polymerase and the epsilon DNA polymerase. One does lagging strand synthesis of nuclear DNA, and the other does leading strand synthesis of nuclear DNA. This polymerase is involved in the initiation of the process and some DNA repair. It also can, in some cases, add an RNA primer. What are some other ways in which DNA replication is different in eukaryotes? Well, eukaryotes have two unique properties to their DNA. One, the DNA is complex to a histone, and two, the DNA is housed within the nucleus. And so the eukaryotic cell has to overcome these two problems. Eukaryotic DNA is packaged, very tightly packaged to histone proteins that form nucleosomes, which are all clustered on each other and so on. And so in order to replicate that DNA, you got to move these proteins out of the way in order to access the DNA and copy it. And so we have to disrupt the nucleosome formation on the parental DNA and unwind the DNA that's wrapped around these spools, move them around. And so these histones that already exist have to be kind of like redistributed on the new DNA. And then we're also going to run out of histones because now we have to split the same amount of histones between two different strands of DNA. So we also have to make brand new histones and add those to new DNA strands. And so there's this process of unpackaging and repackaging and redistributing all of these histone proteins that's coupled to the elongation process of replicating DNA. DNA replication is also confined to the nucleus, and so all of the machinery required to replicate DNA must also exist in the nucleus. So not only are we trying to stuff a humongous genome into the nucleus, but we're also trying to pack it full of polymerase enzymes that will replicate the DNA. DNA polymerase is actually fixed in location, and the template, or the DNA, is threaded through the DNA polymerase. And so DNA polymerases are attached to the inner wall of the nuclear membrane, and then the genome is sort of threaded through it. Now comes the end replication problem. When we have a circular piece of DNA, let's see, we opened it up here. We started the process by adding a primer, and then we had a 3' OH here with which to add new DNA. And we keep doing that and go around the circle until we bump into that primer. Pull that primer out, attach a new polymerase here, and fill in the gap. Boom, no problem. No loss of DNA, no end replication issues. That's because it's a circle. In a linear piece of DNA with multiple origins of replication, we can solve the problem of the gaps in the center, but we cannot solve the problem of the gap at the end. And so remember, there's lots of origins and there's many leading or lagging strands. And so we're going to have lots of little green sections, lots of little RNA primers. They are removed. And then these OH, these 3' OHs here can serve as a template to fill in this gap. This one here can fill in this gap. This one can fill in this gap. And that works all in well for everything in the center. But what about the end? When this primer is removed, we now have a gap left over, and there's no additional template up here with which to synthesize a primer to. And so what do? We just have this big gap. This means that we're going to lose some part of our genome every single time it replicates. So we need to come up with some solution to that problem. And the solution that evolved was the addition of a telomere, or the end cap on the end of a chromosome that doesn't have any protein coding genes and is allowed to degrade over time without any physiological consequence. The enzyme telomerase is responsible for adding on that cap. Telomerase is an enzyme made up of protein and also RNA. And so here's the protein, the yellow sphere, and then there's an RNA template embedded within the protein. The protein will attach to the end of a protruding sequence of DNA, so the very end of a replicated chromosome where there's a gap, and will bind in a complementary way, and then elongate this end, this 3' end, even further. And so what looks like we're doing is we're creating an even bigger gap. But what we're doing is adding a bunch of junk to the end of this, that if it ever gets degraded, it's not a big deal. It doesn't matter. And so that happens over and over until we have this nice protective cap that doesn't represent any protein coding genes, that still has a little bit of a gap. But when that's removed, it's not a big deal. Like, we can lose all of this DNA and it won't have any physiological consequence to the cell. The problem is that telomerase is only active during the embryonic stage of development. And so all of your telomeres are established before you are born. They're all added to all of your chromosomes before you're born. And so that means after you are born, every time your genome replicates, every time your cells divide, you lose a little bit of genome at the ends. This is not a big deal at first because we have lots of N to lose without any consequence. But as you get older and your cells repeatedly replicate, we are going to have a problem where that N gets shorter and shorter and shorter. And eventually the cell will become unstable as we start to chew back protein coding genes. And so telomerase is not active during your life in your body cells. It's only active in certain specific situations. So it's active in cells that have to divide really rapidly in order to perform their function. Those rapidly dividing cells actually do have some telomerase activity. So like sperm, skin cells, and some white blood cells replicate very quickly in order to do their job. And so telomerase is fairly active in these cells, which preserves the genome over time. But most of the time, it's only active in embryonic stem cells. And in fact, it's also an adaptation that was acquired by cancer cells. Cancer cells are cells that replicate over and over really quickly without any check. And so in order for a cancer cell to remain somewhat stable over time, because every single time it replicates, it's going to lose some genome, it has to find a way to preserve the genome so that it can survive. And so one of the ways in which it does that is it reactivates telomerase. And so cancer cells can survive through rapid cell division because telomerase is repairing the ends.